Welcome to Spring LIB.
So here's chapter 9, number 47. It's kind of a long problem, so I'm not going to go ahead and read it, but we just can dive right into the problems. First one is A, describe the advantage of antibiotics to the organisms that produce them. So antibiotics, normally we think of that's how we fight infections. And it's true, they are a competitive tool against other organisms, but also antibiotics are a useful signaling molecule. So a certain bacteria could use antibiotics and release them into their environment. And if it reaches a friendly bacteria, or one that's friendly to the initial bacteria, then maybe it'll prompt that bacteria to create antibiotics as well, or stop creating them because there's enough around. Or antibiotics can go and attack other bacteria that might be competing for similar resources. Now we can look at B. One way that bacteria are becoming a problem is if they're becoming antibiotic resistant. And this problem is saying that even if there's only small amounts or trace amounts of antibiotics in the environment, it can still change the way bacteria are, or it can still cause mutations in bacteria in trace amounts. And even then, it's selecting for antibiotic resistant bacteria because only the ones who can withstand those mutations are able to go on to the next generation, and the ones that can't are going to die off even from these trace amounts of antibiotics. And so, one way that scientists think that this can be bypassed is if we use quorum quenching instead of antibiotics. And what this means is they want to be able to regulate the bacterial expression of genes of bacteria without really selecting for only the bacteria that can be antibiotic resistant. And as we know, quorum sensing is when bacteria can, or is when they send autoinducers out into the environment and see how many bacteria are in their colony or in their population at the moment, and whether or not they're ready to form a biofilm and start other processes. And so if you can quench the quorum quenching, then maybe that'll be able to inhibit certain diseases that bacteria can cause because they're just going to think, oh, okay, we're not ready to make a biofilm. We're not ready to express these genes and perform a certain function. And maybe that can help prevent disease rather than just outright killing bacteria and leaving the ones that can survive the antibiotics.